Hi, and welcome to your first podcast for bisc162. I just want to give you a few suggestions about how to use these podcasts. I recommend using them in the projection mode because I've animated them so that the details of the slides walk in slowly and I tell you when to click. So to do that, at the bottom of your screen or bottom of the PowerPoint window, there's a projection button that you can press and play the slides that way. You can still listen to the audio and you can do that at your own pace. At the end of each of these podcasts, there'll be a series of questions that I'd like you to be able to answer and then bring to class the day after the podcast has been posted, or the lecture after the podcast has been posted. For this one, they'll be fairly simple questions, but I just need to get a sense that you've done them and understand some key concepts at the end of the class. So the first thing we want to do is review the terms that we've used and the parts of the phylogenetic trees that we went over in lecture. There's a lot of new terminology and things can be synonymous, so we want to walk through these at least a couple times. So the first term we're going to walk through is the common ancestor. As you'll be able to see from this diagram, there are going to be several common ancestors, and it's relative to the groups of organisms that you're talking about. So if you're viewing this in PowerPoint as a presentation rather than in the editable form, and you click when I tell you to click, you'll see that a green circle will appear at the base of the tree near the origin of life. This is the first common ancestor we're talking about, and it represents the common ancestor to all four of the groups we've represented at the tips of the tree. This would be bacteria, archaea, plants, and animals. If you go ahead and click again, you'll see that we have another common ancestor that's moved to the right a little bit, and it represents the common ancestor of archaea, the plants, and the animals. If you click again, you can see there's also a common ancestor for just the plants and the animals. This particular common ancestor is one that we can call the most recent common ancestor of tulips and, in this case, mountain lions. So that means that you can have lots of common ancestors through the tree, but often we're looking for the one that's the most recent to a set of taxa. The most recent common ancestor of tulips and mountain lions is shown with the yellow dot. What this represents is not a real living thing. It doesn't mean that there's some ancestor that tulips and mountain lions had, but hypothetically, up until this point, there's a group of organisms or a population that has all the same characteristics that tulips and mountain lions share. Okay, so the next thing we want to review is a splitting event. Splitting events actually happen at the nodes, or where we've just shown you the common ancestors. If you click again, the label for the node shows up. And this is often represents to people speciation. So at this point, we know that they had shared characteristics, but that's about to change. They're about to become different. We represent that by drawing two vertical lines or slightly vertical lines that represent that these two things diverge or become different. To illustrate that they're really different, we draw a horizontal line that we label a branch. So go ahead and click and you'll see the label for a branch show up. And this represents a lineage. So a group of organisms that maybe are exchanging genes. They still have all the shared characteristics, but the difference is in this case, time is happening. So you have time passing as you go towards the right. Okay? This means that all the things along that branch have shared changes. They don't look like their common ancestor anymore, but they're still sharing things as they move along this branch. Finally, when you get all the way to the right and you see the pictures of each of our representative organisms, we call these on the tree a tip, and they're usually represented by some taxon. Taxon is the word, it's sort of a vague term to represent organism, and it can be representative of a whole group like we have here. So for example, there are other animals other than mountain lions. When we talk about many different taxon as a group, the plural is taxa. And usually what happens is these taxa have many new traits. So the reason they're at a tip is that they've accumulated differences, and we know that they're different from one another. You can see that we've represented a new trait with a red circle on this phylogeny. Okay, the most important part of phylogenies are actually the structures that are called monophyletic groups. Monophyletic groups include an ancestor and all of the descendants from that ancestor. So if we take a look at the yellow dot again on this phylogeny, we would include the yellow dot, the tulips, and the mountain lions to form one monophyletic group. That's because you have the ancestor and the tips, or the descendants from that ancestor, included. If you click, you'll see a purple box show up that surrounds the ancestor and the two descendants, and that represents one monophyletic group. Like other parts of the tree, it's all relative. So you also have a monophyletic group that includes the ancestor, the first ancestor back that's green, and that would include the descendants of archaea, the tulips, and the mountain lions. If you click again, you'll see that another purple box shows up surrounding the ancestor in green, that's at the end of the arrow that says node or speciation event. And that also encircles the taxa, or the tips, of archaea, tulips, and mountain lions. Again, if you want to see this labeled with terminology, you can click again and the label for the monophyletic groups shows up. Keep in mind, we use the term monophyletic group and clade to mean exactly the same thing. What they represent is that these organisms have a shared evolutionary history. They have an ancestor. Remember, this represents something that has all of these very similar characteristics, but then they also have become different. But still, historically, they had an ancestor, so they have traits that are very similar. And that shared evolutionary history is to us very meaningful because it can tell us a lot of things about the group. So if you think about something like eukaryotes, we know that all eukaryotes have a nucleus, they have membrane-bound organelles, and they have some other characteristics you can probably remember from 160. And so we know that they acquired these traits a really long time ago, and we call them a monophyletic group because we know they shared those traits, the nucleus, the membrane-bound organelles, before they became different from one another. Last time we were in lecture, we talked about all the different types of data that you can use to build a phylogeny and some of the applications of what we can use phylogenies to do. Today, what we're going to work on in this podcast and in lecture on Monday is how to use the data to draw or build a phylogeny or phylogenetic tree. So in this podcast, I'll walk through how it's done using a method that we call parsimony. This introduction will help you practice so that during lecture on Monday, you can use a new set of data to build your own phylogenetic tree. Parsimony refers to a method that tries to explain that when the best phylogenetic tree is built by the simplest explanation. So that means that the simplest explanation would be the best explanation for how organisms are related to one another. In terms of evolutionary history, what this means is that when two organisms or more share a trait, they share it because their ancestor had it. For example, we know that both tulips and mountain lions have cells with a nucleus. Parsimony tells us that the nucleus would then evolve once, So tulips and mountain lions are descendant from an ancestor that had a nucleus. If parsimony were not the explanation, we might infer that plants and animals developed a nucleus each in a separate process, and that these nuclei are different from one another because they evolve through different processes. That would mean there would be two separate events to form a nucleus, making more steps on our tree necessary. The way that we actually build a phylogeny using parsimony is to collect data on the organisms of interest and compare the traits that they have to one another. The traits that are most useful in building a phylogeny are those called synapomorphies. Synapomorphies, the word is shown in the title of the slide. These are traits that are shared between two or more organisms and are different from what we call ancestral traits. In this case, the term ancestor here doesn't actually mean a physical thing, but it's a trait that the ancestor might have had. And this is a different term than the ancestor that we've been indicating to describe the nodes. So a synapomorphy, again, is a trait that's shared between two or more organisms and is different from what we call ancestral traits. When we talk about being different from the ancestral trait or the ancestor, you need to be given some information about what or who the ancestor is. When scientists build phylogenies, usually they can determine the ancestor by defining a group or taxon that they call the outgroup. An outgroup is a taxon who shows ancestral traits. If a scientist knows a lot about their group of interest and they've been studying it for years and years, they can also pick out a useful outgroup because they know details about traits of their group and usually traits of the outgroup that they're picking. For you guys, I'm just going to tell you what the outgroup is anytime you're asked to construct a tree. It will either be called outgroup as its name, or I'll tell you which taxon represents the outgroup. In the example in this podcast today, I'm going to use bacteria as the outgroup, and I'm going to right away tell you, in this case, bacteria is our outgroup. This means that the traits that bacteria have are the ancestral traits. So, for example, the bacteria group doesn't have a nucleus. So the absence of a nucleus is an ancestral trait. Okay, so we're going to kind of work through two different things here. I'm going to tell you about what the meaning of a certain part of the phylogenetic tree is, but then I'm also going to tell you about how we draw it. So first, if we're going to build a phylogenetic tree using parsimony, the first thing we have to do is find the outgroup. We now know that that's bacteria because I told you it is. The second thing you want to do is to find two or more taxa that share traits that are also different from the outgroup. When you look at a table of data like you see here, this means you're looking at columns, you're not looking at rows. So within a column, we will actually look at the whole column together and see which group, Archaea, Animalia, Plantae, actually has those characters. So what we're actually looking for is a type of character that's called informative. If you click now, the term informative will show up and it's pointing to one particular cell, but it really refers to the entire column. The column the arrow is referring to is the one called protein X. Protein X itself is what we call a character. Whether or not protein X is present is the trait it has. So in this column of protein X, you can see that bacteria do not have protein X, so the ancestral trait is to not have the protein. However, you can see Archaea, Plantae, and Animalia, or Archaea, plants and animals, all have protein X. This means that when compared to bacteria, the presence of protein X is a synapomorphy. This is because three groups, animals, plants, and Archaea, share the trait, and it's different than the bacterial outgroup. So in order to be informative, it has to be a synapomorphy, it has to be shared and different from the outgroup. Remember that shared implies that two or more taxa need to have the trait for it to be informative. Again, this means that when we go to build a phylogeny, we need to look at data in columns. Here there are 5 columns, or 5 characters here, cells, protein X, nucleus, organelles, and chloroplasts. When looking at this data table, the cells are filled with simple yes or no data as our traits. Yes means the group of organisms has the characteristic, and no means the group of organisms does not. Now that you know what an informative trait or character is, you can identify any traits that are not informative. Since you're looking for traits shared between two or more groups, and they must be different from the outgroup. So what we're going to do is I want you guys to take a look at this table and try to identify which of the characters or which of the traits are not informative. Go ahead and pause here to try to find the informative characters. All right, you should have found that both the cells column and the chloroplast columns are uninformative. The other three are informative. Here's why. All groups of organisms have cells, so they are not different than the outgroup. Only one group has chloroplast, so there's no sharing between taxa. What this means is this doesn't help us figure out who's most closely related to who. Right? It's true that everybody has cells, but that doesn't tell us whether bacteria is more closely related to archaea or animals. All it tells us is they all have this characteristic. So just because these characters are uninformative, it doesn't mean they're useless. It just doesn't help us figure out how each organism is related to one another. So the three remaining columns, protein X, nucleus, and organelles, are what we have as our informative characters, and this is because they're shared between two or more taxa and they're different than the outgroup. All right, so now that you understand informative and uninformative characters, we can actually work at how to draw the tree. This part can feel tricky because you're always worried you're doing it wrong, and that's okay if you do it wrong a couple times. You just have to start again. So I encourage you to bring a pencil to class on Mondays so you're comfortable with trying to draw out these trees. I only use a pencil because I tend to make mistakes when I first do my drawings. One of the things other people like to do when they get the table of data is once they've found their informative character, circle all the individuals who have the shared derived trait. So it really stands out and it's easy to see in a table. It's harder to do on PowerPoint. All right, now take a look at your informative characters again. Of the columns, which of them is shared by the largest number of taxa? Well, you should have identified the protein X column. 3 taxa share that, Archaea, Animalia, and Plantae. The only group that doesn't have protein X is bacteria, your outgroup. I've already told you this is the outgroup, so what protein X tells you is that bacteria differ from the remaining taxa because they do not have protein X. If we want to start drawing a phylogeny, we already have a bunch of information just from this. So if you go ahead and click, we can see what the process of what it's like to start drawing. First thing I always do is to draw a branch that represents the origin of the group. So it's a branch, so it's horizontal. And if you click, you can see I've drawn a horizontal black line to the right of the table. Anytime you get a horizontal line like that and it's not at the tip of the tree, you'll also have a splitting event. So you can also go ahead and split that branch into 2 vertical lines. So the way that we show this, remember that vertical lines are showing a splitting event, and we can show that by drawing 2 vertical lines off of that original branch. So if you click, those 2 vertical lines should show up. We also know right at the start of any of our phylogenies what our outgroup's going to be. In this case, it's bacteria. So when you get the outgroup and you know what it is, it's good to always start by drawing that one in right away. It doesn't matter whether you put the outgroup at the top or the bottom of the tree, but for this one I put it on top. You can see a branch appear at the top when you click, and then its label, and on the next click you can actually see the picture appear. If you wanted to, when you're doing your own trees, you don't have to put a picture there, you can just put outgroup at the end at the tip of the tree. Now, we also know that once our outgroup is in place, we also know that there's going to be a vertical line coming off of cells that should have already appeared. But At the vertical red line, what appears red, you should also get a horizontal branch, a short one, kind of like you drew at the beginning of the origin. So if you click, a short red branch should appear. We also know at the end of that red bar, you'll get a splitting event. That's how we draw these trees. You have 3 more taxa to add, so you know it's got to split at least one more time because you have many more taxa to add. So again, if you click, you'll get a set of vertical lines that come off of that red branch. All right, that's a lot of practical detail about how to make yourself comfortable in the drawing steps. But now we need to actually apply the data in the table to make this tree. We know that archaea, animal, and plants are more closely related to each other than they are to bacteria. This is based on the fact that they all share protein X. This means that on that first vertical red line that comes off of your original black line, this is when protein X evolved, sometime before we get that split at the horizontal red branch. If you click, you can see how we show this on a tree. We use a circle to indicate the trait, and then we can actually label it. And it means now that anytime after that circle, every taxon represented still has protein X. We've now taken all the information from the protein X column, and we can't use it to inform us anything more about our relationships. So when you have your own data table and a piece of paper, you can check it off so you don't keep going back to that column. Okay, so now we've taken the information from protein X, and now we have to figure out how archaea, plants, and animals are related to one another. And there are really 3 options. Archaea and plants could form a monophyletic group, archaea and animals could form a monophyletic group, or plants and animals could form a monophyletic group. To figure out which is true, we go back to the table again. Take a look at the 2 remaining informative traits, the nucleus and the organelles. Which taxa have synapomorphies? You should see that plants and animals both have a nucleus and both have organelles. These 2 traits tell us that plants and animals share more traits with each other than either plants or animals share with archaea. And this also tells us then that plants and animals form a monophyletic group. It also tells us that these traits are not present in archaea, and so they evolved sometime before plants and animals became different from one another. But after archaea is no longer in their group. All right, if that's the case, then how do we draw it? Well, we know that the 3 taxa are monophyletic because of protein X, but we also know that plants and animals form a monophyletic group that does not contain archaea. So we draw a horizontal line from the vertical red line and put archaea at the tip. Go ahead and click to see that. Then at the blue line, we draw a short horizontal branch that indicates a lineage existed before plants and animals became different. And again, if you click, that will show up. We can put the traits on the tree, the nucleus and organelles, and they appear on the click. Okay, now this is where people get a little frustrated. Plants and animals are not the same thing. We know that. Mountain lions and tulips are not the same. But we have no informative traits left, and sometimes even no uninformative traits to tell us how they're different from one another. So should you put them on the same branch? Is there any information to make them split from each other? Maybe they do go on the same branch. No, it doesn't make any sense. Every taxon that you have in your table will end up at its own tip. You know tulips and mountain lions are different species. So when you think about drawing this last bit, even if you don't have information, you know that each taxon gets its own tip, so you'll always split them like you did with the other splitting events. This means you'll draw vertical lines. They should show up as solid blue when you click, and then 2 horizontal branches leading to the 2 tips. And if you click, you'll see them appear. Now in this case, I put plants on the top and mountain lions on the bottom, but you could do it the other way. The information in a phylogenetic tree tells you which taxa form a monophyletic group. If you were to switch tulips and mountain lions, it doesn't change that they're still monophyletic. And look, now you have a tree telling you which taxa are monophyletic and which traits inform you about the relationships. And you have all of the members, all the taxa you were interested in looking at the tips of trees. Okay, one more thing about this. Remember, I said that uninformative traits aren't useless. You can put those on the tree too. The way you would do it is to indicate with a circle that cells are found in everything. So you label that on the first branch that was drawn, the black one. And then you can add chloroplasts to the branch leading to plants. If you click, these appear in order. So in general, we've just built a phylogenetic tree, and the way we did it was to first, in step 1, find the outgroup and draw it on the tree. Step 2, find the informative characters, and it might be really helpful to cross off the uninformative. And then find the trait that's shared by most organisms, use that to label the monophyletic group, draw that on the tree, and then find the traits that are shared by the next most common until you run out of taxa. One thing to remember about phylogenies is they are relative depictions of evolutionary history, so they only show the relationships between the organisms of interest. What you see at the bottom of this slide is a picture of 2 eukaryotes, the mountain lion and the tulips. This is the same relationship from the previous slide, but instead of the mountain lion being on the bottom, they're on the top, and the tulips are on the bottom. They still share a common ancestor and therefore still form a monophyletic group. However, when we show just one image to represent plants and animals, we know to expand those clades. Animals are not just mountain lions. In fact, mountain lions aren't even the only cat. So when we get really detailed into phylogeny, we can expand these into additional monophyletic groups. So, for example, if you click now, what you'll see is the mountain lion's image will go away. You'll see the formation of a new clade, so you'll get an ancestor and 2 descendants. And then if you click again, you'll see the images of a domestic cat and a mountain lion appear. What you get from that is mountain lions and domestic cats have a common ancestor. They form a monophyletic group in this image. But you could even expand this further. We know that there's not just one type of domestic cat, so we could expand that into having more than one domestic cat. So if you click again, you'll see that the domestic cat represented a larger group of organisms, in this case on the top a black cat named Vinny, and on the bottom a picture of another cat named Matilda. These 2 cats are both domestic cats, they share a common ancestor, and they belong to a clade themselves. So this process can be repeated along each of the clades. Now, if you wanted to just pretty up the diagram a little bit, and you continue to click, you could expand the lineages for the mountain lion and the lineage for the tulips and extend them out so they're all even on the right side of the page. Keep in mind that by doing that and extending it to the end of the page, you're showing that these things all exist at the same time. The reason that I show you that the tree can be expanded at every tip that we show is that there are a lot of different species, and that's why we look at biodiversity. As you go through the lab, you're going to look at representatives from many different groups of organisms. If you've had lab already, you know that you'll see some examples of cyanobacteria, you'll see some different shapes of bacteria, and maybe even some of the more pathogenic bacteria. If you look at this phylogeny and you're interested in where that would go, you wouldn't just call it the tip called cyanobacteria, but you'd put it in with that clade. So as you're studying the material from the lab, in order to organize what you've looked at, you can add each slide or each organism that you look at to the tips of the tree. This phylogeny shown on the left is in your book. It's at the end of the book. It's also on the ebook, and you can zoom in on this and kind of manipulate the different ends of the branches if you want to. This is a really good way to help you organize the taxonomy, and I will put together a few other slides in a few days to show you how you might add those things on as you're studying. Since this is one of the ways that we do organize using a field called systematics, we're going to expect that you can read a phylogeny to find the organisms that you've looked at in lab. In class on Friday, we're going to actually build a phylogeny using data like we did in the earlier slides. But one of the important things about phylogeny is it can help you organize all of the data that you get as you're doing lab. So I want to walk through an example of how you might look at a phylogeny and use it to help you think about what you saw in lab. So what you see at the top of this slide is just the cut off top part of that large phylogeny from the previous slide showing the bacteria clade. In lab, you looked at bacterial shapes, but you also looked at things that have specific names, and they belong to the group called cyanobacteria. So if we look at this phylogeny, or this subset of the big tree of life, and try to find the clade that includes cyanobacteria, but is still a monophyletic group with a lot of other things. What you can see is that cyanobacteria, it's number 43 on the numbering system, shares a common ancestor with 3 other groups of bacteria. One of them is proteobacteria, another is spirochetes. The most recent common ancestor of cyanobacteria, proteobacteria, and spirochetes is actually quite a few nodes back. So if you click now, you'll see a blue box come up and surround the monophyletic group that includes cyanobacteria. If you click again, what you'll see is sort of an expanded phylogeny showing these 4 groups of bacteria. Now in lab, you looked at 4 different species of cyanobacteria. So if we wanted to represent this on the phylogeny, we could actually delete cyanobacteria. So go ahead and click, and what you'll see is the name cyanobacteria disappears, and so does the one below it, but that's just so that we can see a little bit better. And we can do exactly what we did when we added Vinny and Matilda to the tree a couple slides back. Alright, so click again, and what you'll see after the text disappears is another clade show up with 2 branches. Now in lab, you learned that there were 4 different species of cyanobacteria that you're going to use. So we actually need 4 tips within this clade. Since you don't know how all of these cyanobacteria are related to one another, you can just draw it so that they all belong, or all share a common ancestor and belong to the same clade. So if you click again, 2 more branches will show up. And then you'll also see that all 4 of the taxa you looked at in lab will appear in the text. So that's Anabaena, Nostoc, Glyocapsa, and Oscillatoria. Then, if you want to summarize what these examples represent in terms of taxonomy, you would say all belong to the clade Cyanobacteria. So if you click, a green box appears surrounding the ancestor of all 4 and the name showing that this is Cyanobacteria. So what this phylogeny then does is it shows you that examples that you look at in lab can be representative of a monophyletic group known as Cyanobacteria. If you do this for all of the things and you keep track of what you've done in lab using the tree of life, it becomes less like a list of organization rather than actually a big picture of what you've seen.